Many experiments were conducted in search of the genetic material in order to prove which is the genetic material, whether it is a DNA or RNA or the proteins which is acting as the genetic material. So let us look into the experiments, the search for the genetic material. The first experiment is the Griffiths transforming experiment, the Griffiths transforming principle experiment. And the name of the scientist is Frederick Griffith in the year 1928. And his experimental material was Streptococcus pneumoniae. It's a bacteria which causes pneumonia. And along with Streptococcus pneumoniae, mice was also used. So the two experimental material were the mites and the bacteria which causes pneumonia, namely Streptococcus pneumoniae. When Streptococcus pneumoniae, they were grown in culture plates, it was observed two types of colonies were formed. The two types of colonies were the smooth strain and the second type is the rough strain. What is a smooth strain and rough strain? The smooth strain are the virulent forms. That means these are the strains which cause pneumonia. And why it is able to cause pneumonia? It is because they have a polysaccharide mucus coat. In this we can see this is a smooth strain and this is the rough strain. What is the difference between the two? It is based on the outer surface. The outer surface of the smooth strain is smooth in appearance. It is because of the presence of a polysaccharide mucus coat which gives them a smooth appearance, and it is this smooth polysaccharide mucus coat which causes virulence or it causes pneumonia. Whereas the other strain is the rough strain, and this is the non-virulent form. It does not cause pneumonia. Why? Because it is lacking the polysaccharide mucus coat and thus it does not cause pneumonia. So the two types or two strains of Streptococcus pneumoniae are the smooth strain and rough strain. Smooth strain, they are virulent, whereas rough strain, they are non-virulent. Why they are non-virulent? Because they do not have the mucus polysaccharide coat. Whereas the smooth strain, they have the mucus coat, polysaccharide coat, which gives it the virulence property. So there was a series of steps in the Griffiths transforming principle experiment. In the first step, he injected living estrine bacteria into the mice. And he found that the mice died of what? What does the estrine cause? It causes pneumonia. So the mice died of pneumonia. So in the first step, live estrine was injected into the mice and the mice died of pneumonia. And in the second step, we can see he injected living estrine bacteria into the mice. And will the mice die? No. The mice will live because estrine does not have a virulent property. It does not cause pneumonia. So living estrine injected into the mice will live. And in the next step, he killed the estrine by heating them. So he heat killed the estrine. And then he injected the heat killed estrine into the mice. And he observed that the mice lived. The mice lived. When heat killed estrine bacteria was injected into the mice lived. In the fourth step, he mixed the heat killed estrine plus the R strain which was living. So the dead heat killed estrine and living R strain was injected together into the mice. And he found that the mice died of pneumonia. The mice died. And he also observed that living estrine cells were found in the mice. He discovered in the dead mice living estrine cells. But these estrine, they were actually heat killed. But when it was mixed with R strain cells and injected into the mice died of pneumonia, and Griffith discovered living S strain cells or S strain bacteria in the mice. Griffith concluded that something was transferred from the heat-killed S strain to the R strain, and this something is called as the transforming principle. And this transforming principle, it enabled the R strain, which is non-virulent, it does not have the mucus polysaccharide coat, to synthesize. This transforming principle, it enabled the R strain to synthesize smooth polysaccharide coat and thus become virulent. And this is due to the transfer of the genetic material, and this is called as the transforming principle. This is called as the transforming principle, and that is the experiment, Griffiths transforming principle experiment. And this was the first experiment in search for the genetic material.